data analysis part, because data analysis is very critical at the end. And many of the students or the participants of MOOC, if they want to analyze the data of mass spec at their home or using their laptops, how they can do that. So there are some new open access software coming up, which is like the Frag Pipe or MS Fragger, which is quite easy to do that in your PCs. So the next session is going to be talking about data analysis, and where our teams are going to show you how to take the your DD or DIA files from the mass spec and use this open access software, which is the MS Fragger. I will uh, just show you, like, how the software logo and things like that, so that if you want to make yourself familiar, you can do that also. But idea, this software is done from one of the academic lab, and here the things are so fast that you don't need too much RAM from the clusters, and you can directly use with the limited RAM and the computing power of your laptop, and directly analyze the any kind of workflow of DDA and DIA. So Amit, please share your screen and just briefly show the frag pipe data analysis.
Okay, thank you, sir. Yeah. So, yeah. So I guess you can see my screen, and here you can see the interface of the FragPipe software, the FragPipe version 23. Now, let me just give you the very minute brief of what is the interface looks like. So this is the basic interface. And one of the most important thing, whenever you are going to analyze any kind of raw files in the FragPipe, you need to set the path you need to set the path. So here you can see I have kept two different raw files here. That is came up from a rat, that is based on some rat tissue samples. Now, in case of this kind of analysis, we need whatever the path you are using in this entire path, in any name there shouldn't be any space. So you can see there is a name NPTEL 2025. There is no space. There is an underscore, and even the output files you don't need to keep any space here. If you keep space, the software will stop analyzing the samples. Now, the very first thing is that you need to first upload the data files here. So here is the workflow tab. So yeah, basically here is the workflow tab. We'll add files. Now you will go to your folder. So basically these are two raw files you need to upload. You select this, and you can see these files are uploaded. These are DIA datasets. You need to set DIA. Now you need to set the experiments here. So and also and if these are bioreplicates, you can also set what are the replicates. It's a control sample or a disease sample. You can also set it. For now, I am setting this for consecutive. Now you need to also upload the different workflow. So it's a DIA file. You need to upload the DIA spec quant. Okay. Now now you need to keep certain RAM and at a certain parallelism. So if you want to try out, please see what is the code of your system and set the parallelism based on that. Now then the next thing is the database. So we are using a RAD database. So basically the entire database you can upload with the help of UniProt. Now you can just browse it. I have already downloaded the database here, the RAD database. I'm going to upload this database and select it. I can see this is uploaded, and you will add decoy. So decoy is basically a completely opposite of the entire FASTA file of the RAD proteome. So basically you have to find out any kind of false to remove any kind of FDRs there. So this is basically a non-existent database that will basically try to match with your original databases and remove the false positive sequences of the proteins. It's taking some time to upload database. So after the database is uploaded, you can go to yeah. So you can go to just MS Fragger and you can set or what are the different proteins that you are like. I am selecting a trypsin. you can also selective lyse C or anything. You can also change the miscleavage. Then we can go to validation. So in validation, this peptide spectrum validation is important to see any kind of PSM is there. And then you'll go to run, and you'll set the part that where you actually put the output files there. Yeah. So we'll just keep the directory set here, and you will run. Yeah. So as you can see, the stuff file starts running.
All right, so great. Thank you, Amit. So, what students can understand here that you've seen how quickly the entire process could be achieved, and this file will take maybe good half an hour trying to run. But earlier, the same files used to take maybe several hours to day time to run, but now it can be done in just very short time in less than an hour. So, another software which will now show is Mascot matrix science, which Ankit will briefly walk you through and show you, like, if you have a very simple mixture of the protein, you want to identify their sequences and you want to know what is the bigger hit, how to utilize that using the Mascot software analysis. So you'll do that, Ankit.
Yeah.
Amit, you can unshare, yeah. Ankit.
Hello everyone. Yes. Yeah. So I'll show the interface for the mascot. So we had talked about a lot, right? Like getting the peptides and the mass spec. So now how we had identified, so PracPipe was one of the software. So in the matrix science mascot, so this is very user friendly and easy to use software for files that are not so much complex. So what we can do is that, so first of all, you can just register here with your email ID and give the search title. So here we are using two databases. So since if we are not aware that what is the particular sequence that contains from which organism, we can give all the Swiss prot and some contaminants which are general happens when while you are doing the kind of digestion and other processes like keratin and all can come from different sources. Then taxonomies, we can give all entries. Enzyme, you can add like trypsin. Then we are not adding any kind of quantitation or cross-linking as such. And then you can simply add the fixed modification like IA and those for those things which we had already shown in peptide digestion. And then here we can go to do like oxidation, but if you want to have any ppms and all, you can add it. So then you can keep it all default with all the standards that are present, that are community based standards. Then you can choose your data file. So it is taking in the .mgf, that is mascot generic format. I have already uploaded it, and then you can simply start search. Okay, so it will start running, and it will give us the result, and you can see so the y ions and b ions and those things that we talked upon, like getting the actual peptide. So here you will get that after adding that the identification of the peptides for a particular protein, you will see that for this particular protein this number of peptides have been identified. And now if you go and click for any particular peptide, so you will get the fragmentation spectra or pattern as well, like the ones that I was telling
All
that
right.
for the
Thank you, Ankit.
peptide.
So this is a very quick one, but at least students got a feel of it, how to do different software for analysis. You can stop sharing your screen. So just in the final minute or so, students have been also asking us in which way the proteomics is really, getting more and more transformative using the new mass spec technologies. So what I'd like to mention to you that our ability to really look at the proteome from the very small amount of protein, for example, as we just show you in the timsTOF, using 200 nanogram or 3 nanogram amount of peptide, one could now look at a complex proteome to more than 8,000 and 10,000 protein. Now we are actually started to reach towards understanding some key biological questions. Can we now do single cell proteomics from the single cell? Can we look at the cross links of the membrane protein and trying to look at more of the druggable targets? Can we look at even the imaging of the cell and use the spatial proteomics now as the new way of looking at where all the our major protein changes are happening? So there are many new workflows are emerging, and thanks to all these new instrumentation, new technologies of mass spec, which is now making the proteomics to a very, very unprecedented scale, which was not possible earlier. And now we can see different type of applications are going to be really quite important for the clinical as well as biological applications. I hope students who are taking the MOOC courses on proteomics and proteogenomics are keeping the pace, the new technologies that are happening, and you're reading them well. And if you have additional questions and queries, please do get in touch with us in our separate Telegram groups or in the chat, like different type of chat which we do on your forum. We are quite available, and do make this kind of forum as a more opportunity for your live interactions with us. I think some of you have also asked us whether you can get some opportunity for doing workshop in our lab. Of course you can, but you have to be the top performers in your course. And then we do give opportunity as an internship or some of the workshop we conduct pretty much free of cost for the student to come and learn those technology directly in our lab. But that we cannot give to everybody. That has based on the selection process to the top performer students. So wish you all the best for your exam for the proteomics MOOC course students, and proteogenomics students. We will have one more session with you in the next month. Thank you very much, everybody, and goodbye for now. Thanks the TAs for all the live session, very nicely interacted and well coordinated. Thank you, team.